Whenever you're ready, please.
Let's see. This one doesn't work. Okay. Well, thanks for the introduction. This is difficult for me because I'm the only thing that stands in the way of dinner. So, and I start with an apology for those of you who have seen some of this material before, but it's hard to figure out how to present it without presenting the fundamentals. So I start with what we don't know about water, and all of you realize that there's an awful lot that we don't know, but some of the things are so common, and there's common enough that we hardly even think about them. The questions are too obvious to ask. So one question is, if you look at a cloud is suspended in the sky, and cloud is made of water. Water is heavier than air. Gravitation pulls it down. So what keeps the cloud floating in the sky? Maybe you haven't thought of that question. This is an unanswered question about water. Another question related to this is, you know, if you see a picture like this, the water is evaporating from everywhere, from here, and from here. How is it possible that you can get one distinct cloud that sits there? Why shouldn't there be a cloud that extends fully over the surface of the water? How could this be? Another one is water meeting water. And as you know, when water meets water, the two coalesce. But here is an example from our laboratory of water droplets being dropped from a pipette onto a surface of water. And you can see it doesn't dissolve instantly. How come? What's going on? And how come we don't know the answer to it? We saw a picture earlier about a water bridge. And just think about it for a moment. You put two beakers lip to lip. You put one electrode in here and one in here. And you put a high voltage between them. And here's what happens. So a bridge forms between the two. And then if you move one beaker away from the other beaker, the bridge sustains itself. And it sustains itself for a distance of up to 3 or 4 centimeters. And it does it essentially indefinitely. Have you ever seen water that behaves this way? What's going on? And how come we don't know the answer? Another example comes from Professor Shogo Ueno from University of Tokyo. Pardon me. Now, he took a superconducting magnet, which you see back here, placed behind a trough of water, into which is put some red dye, and you can you'll see in a moment why he put red dye here. So when he turns on the magnet, the Red Sea splits enough for Moses to walk across it. What's going on here, and how come you don't understand it? Or maybe you do, I'm not sure, but I bet you don't. We originally were studying muscle contraction and got started some years ago when I met Gilbert Ling. And Gilbert Ling came from China. He was one of the first group of 3 people to come from China after World War II, selected from all of China, right? And Gilbert, and among the 3, they all thought Gilbert was the cleverest of all. And another guy, one of the 3, Yang, won the Nobel Prize in Physics, so this was a rather distinguished group. I think Yang is equally known for his work in physics, and also the fact that when he was almost 90, Gilbert's 99 now, when he was 90 he married someone age 30 approximately. So he became famous for that too. Anyway, Gilbert had written, by the time I met him, 3 or 4 books, maybe even 5, on water inside the cell. So you know, we think of water inside the cell, most of us, as sort of like this. And Gilbert said, no, it's not true. It's not like this. The water molecules are actually lined up kind of like a crystal. And obviously this was controversial. I gave one of his books, which his books are not easy for ordinary people like us to read, but I gave it to some of my postdocs and students, and every one of them said, this guy is on to something really important. And if true, obviously, if the water molecules inside the cell are organized in some way, this is vastly different from the current model that virtually every biochemist and cell biologist uses. So I took it upon myself, I got so interested, and I took it upon myself to try to make clearer some of the messages that came from Gilbert Ling, and I wrote this book in 2001 to describe water inside the cell and the critical, absolutely central role of water, particularly this organized kind of, or structured kind of water in cell biology. I thought the book was not really much of a success, but obviously it turned out to be really important. As you can see here, this is, by the way, the child of Zoka. Okay, I thought you. So selected from all the other books. Excuse me. One of the main messages of Gilbert Ling's work, and a message from the book, is that what happens inside the cell is that you've got proteins which have mainly hydrophilic surfaces, and next to these charged hydrophilic surfaces are water molecules, shown here as dipoles, and unlike physical chemists who generally argue that there may be one or two molecular layers of ordered water next to these charged surfaces, Gilbert said, no, it's not true. The evidence shows that these can line up for many, many molecular layers. Eventually, they give way to randomness because of thermal motion, Brownian motion. But he was arguing for dozens or even more layers of this kind of organized water inside the cell. So we became, I'm sorry, I need some health-promoting water. We became interested, and we were looking for a model by which we can study this experimentally, because it seems so intriguing. I mean, if it's really true that inside your cells and my cells the water molecules are lined up, this is something that is really important to pursue. So one of the characteristics that we understood was in the region where the water molecules were ordered, it's kind of like a crystal, and we know that crystals tend to exclude particles and molecules, otherwise it wouldn't be pure crystals. So anything that's inside gets pushed out, just like ice. When ice forms, it pushes stuff out to give you pure crystals of ice. So we were looking for an experimental model in which we could see something like this, and we found one. And so I'll just illustrate. This is one of the first experiments we did. This is a chamber sitting here, and inside the chamber is a gel. And here's the boundary between the gel and outside the gel. And we put water inside of it with particles. And these particles are microspheres, approximately nominally 1 micrometer, latex spheres functionalized in various ways. And we noticed right here that there was a region that contained no microspheres. And we thought maybe, possibly, this could be a region where the water is actually ordered. You know, we had no evidence for it, but this seemed like a possibility. And if you watch the video of how this works, you can see that the region that we're talking about grows and pushes the microspheres out until with this particular example, this was a polyvinyl alcohol gel up to about 50 micrometers. So this looks like a really large region, unexpectedly large, because if you think of the number of water molecules that would have to be lined up to give you this, we're talking huge numbers, way beyond what Gilbert Ling and others have suggested. So a colleague in Australia said, oh, you know, you see this so often, you should give it a name, because apparently nobody else had identified it. It turns out in 1970, some physiologists from Japan studied gels, including the lens of the eye and artificial lens, and they found almost the very same thing, 1970. So it was not unique after all. So suggestion was call it something, and the obvious label is exclusion zone or EZ because it excluded. So it became the EZ or the exclusion zone, and it's easy to remember. So this has some advantage, except it doesn't work in Europe, and I think it doesn't work in Japan where it's EZ, or is it? I'm not sure. Are you EZ or EZ? Well, whatever. Okay, so easy to forget. Yeah. So another example, and one we've come to use quite a lot, is a piece of Naphion. Naphion is a polymer. It has a Teflon-like backbone, but with sulfonate groups that confer charge. So you see, we put it at the bottom of the chamber, and you see much the same thing. You see this region right next to it that excludes the microspheres, and if you watch it over time, it builds, and in this case, it builds to typically 400, 500 micrometers. It's big enough, a half a millimeter, that you don't even need a microscope to see it. You just look at the chamber and you can see that there's a region that has no microspheres. So we've come to use Naphion quite a lot for experiments because it's so easy, despite the price of these sheets of Naphion. They're really convenient because you just cut it into whatever shape you like and examine it. So quite a few people have gotten interested, and many people have at least seen it and confirmed the existence of these microsphere regions. This was constructed about 5 years ago, and since then there have been many more. So what I want to do, getting to the basis of what this is all about, is I want to answer 5 questions. The first is the exclusion zone phenomenon general, or just those couple of slides that I showed? Does it really arise from the ordering of water molecules, as Gilbert Lin had suggested, or something else? Can the ordering of water explain those first few slides that I showed you, which we couldn't really understand? And next, of course, you need energy to create order. Where does the energy come from? It was not at all obvious to us from the start. The answer turned out to be really simple. And might these findings apply in general, and especially what I'll cover today is for your health. And I also want to touch on the issue, when I talk about the water ordering, on the issue of water memory, which is a topic that interests many people. So first, the issue of generality. So what kinds of surfaces generate this? We studied many gels, hydrogels, various polymers, biological surfaces, and even monolayers on gold, which was discussed earlier. And we found that if the surface is hydrophobic, we find nothing like this. If the surface is hydrophilic, it turns out to be very general. Not every hydrophilic surface shows it, but many, if not most, show it, especially with gels, with hydrogels. We've tried, more than a dozen, and they all show this kind of exclusion. Now, the next question is, what's excluded from the exclusion zone? And we studied a range of sizes of particles and molecules, from big particles to smaller particles, small particles to viruses, bacteria, down in size to macromolecules like proteins and such, all the way down to a molecular weight roughly 100 or so, 100 Daltons and smaller. And it's a very general phenomenon. Not everything is excluded, but most of the substances we look at are excluded from the exclusion zone. So in terms of generality, I summarize that many hydrophilic surfaces generate these exclusion zones, and many solutes are excluded. Okay. Now, next question, number 2, is this zone physically different from bulk water? I've alluded to the possibility that it might be different, but I provided no evidence. Well, over the years, there's been quite a lot of evidence that the exclusion zone water really is physically different from bulk water, and I'll just list. I'm not going to go over all of these, except I want to deal with one of them because it's mostly published, and I want to get on to more interesting phenomena based on this. So the first is that the easy water molecules are more constrained than ordinary bulk water molecules, and we used NMR for that. The molecules are more stable. They generate less infrared energy than ordinary water. We found that the easy has typically a negative charge, not neutral. And I want to go back to this one because it's a surprise. It was a surprise for us. And also it has important implications. It absorbs light at 270 nanometers, which ordinary water doesn't do. It's more viscous by a couple of orders of magnitude than, than bulk water. And Japan is not good for my health. The easy molecules are aligned. This was shown by Nikolai Bunkin in Russia. And the molecular structure is different, and also a couple of Russians have shown the optical properties are different. They measured two different guys from Moscow who didn't know each other. One of them passed, so I guess they're not going to get to know each other. They reported about 4 years ago that the refractive index of the EZ was up to 11% higher than that of, they got the same number, and one was dealing with a biological system and one with a physical system. So these are 8 of the differences. There are more. I didn't want to list them because of taking space and time. So what about this negative charge? This really puzzled us. So what did we do? We took a microelectrode, a glass electrode that tapers to a tip of 1 micrometer or less, invented, by the way, by the same Gilbert Ling I was talking about, who should have gotten the Nobel Prize for even just for that invention, because it's been used so much, and even by the people who are using patch clamps to study channels and such. But anyway, we took one of those electrodes, a pair of them. We stuck one in the exclusion zone and one way beyond in some other region to measure the potential difference, and we found that the exclusion zone was negatively charged. And when we were in the lab, this was a real surprise, because the experiment, you have a chamber and you take some water and you pour the water, and the water is neutral. So how is it possible that you start with a neutral water and you get a big region that's negatively charged? It doesn't make sense. So we weren't sure of this result, and a guy, my friend Felix from Russia, was in the lab at the time, and I remember he made a phone call to his wife in Russia, hello honey, or whatever it is, the Russian equivalent, I don't know. And he said, try this experiment, because she had gels and she had microelectrodes. And so it was really easy for her the next day to try the same experiment, and confirmed. So got the same result. And we tried it first with a gel, and then we tried it with Nafion, and we basically got the same result. But still the question is, how is it possible, right? Because you start with neutral water. And what we thought is somehow the water molecules are breaking up into a negative part and a positive part, and the negative subcomponents, OH minuses, are somehow gathering together in this region to give you the negative. And if that's true, if that were true, then you ought to find the equivalent positive charge somewhere. And if you can't find it, then your hypothesis is all wet. Well, we found it. I'll show you in the next slide. What we did was we took a piece of Nafion, and the exclusion zone is right next to it with negative charge, and we added pH-sensitive dye, same as litmus paper, but the dye, it's in soluble form, and it changes color depending on the pH. So we wanted to see whether there, in some region, that there were a lot of protons to equal the negative charge that we measured with the electrodes. And this is a result. So here's a piece of Nafion, and here's the exclusion zone. By the way, the dye doesn't penetrate. Dyes, chemicals whose molecular weight is about 100 or so, 100 Daltons. So that's why it's clear. I think it shows clear on the screen. So this has negative charge, and the region beyond this color, this red-orange color, is pH 3 or less. So it's just filled with protons. See, so So it appeared to us that that it's really true that somehow the water is splitting into negative and positive. And this would not be a surprise because in the first step of photosynthesis, that's exactly what happens, that the water molecules get split into positive and negative components. So there's good precedent for that. Well, just to be sure that we weren't deluding ourselves into thinking something that was not necessarily true, we put two electrodes in here, one in the negative, one in the positive, and connected them with a resistor. And if the charges are really separated, you'd expect current flow from plus to minus. And this is one of our first experiments to demonstrate this. Kurt Kung, who's here, has taken this miles ahead to demonstrate some practical consequences. But here is current versus time. And so the current starts at a high value and diminishes to a plateau. And the plateau is not zero. Zero is here. So you're getting steady current flow from the positive region to a negative region, basically confirming that the charges are really separated. So what we have, what I've shown you essentially, is you've got a charged battery in water. So this is a hydrophilic surface. This is water. And the EZ sitting right here is negatively charged. And you've got protons beyond. This is kind of unexpected, but that's what we've found. So to summarize so far, there is a liquid crystalline region where the molecules are ordered some way. And the region is not neutral. It's got negative charge. It excludes solutes profoundly. And there's some hint from the list of evidence there, I'll go back to that in a moment, that the dipole model that's shown here, with plus minus, is not correct. A very simple reason why one hates to admit that your own hypothesis and such is wrong, but we came to that conclusion. And this array of ordered water molecules may extend very far from this nucleating surface. And by very far, if you do the arithmetic based on the size of the water molecule and the size of the exclusion zone, we're talking up to millions of molecular layers, not 2 or 10 or a few dozen, but up to millions. It was suggested many years ago by the famous physical and colloid chemist Sir William Hardy, that there's got to be another phase of water. So we all think of the solid, the liquid, and the vapor. But he said there are too many anomalies of water, things that don't fit this paradigm, this simple paradigm of ice, liquid, and vapor. So he suggested some sort of intermediate phase. And a lot of people along the way have been discussing the possibility of another phase of water. And I'm not sure if what I've shown you is maybe indicative of a fourth phase of water, but we tend to call it that. We tend to call it a fourth phase of water. Now, why am I suggesting that these simple dipoles, plus minus, lining up, is not correct? Well, it's because we measured that this zone was negatively charged, but dipoles are neutral. Plus minus gives you neutral. So if you could stack dipoles from here to Kyoto, and you'll never get negative charge. And so it's very simple logic that this lineup of dipoles simply doesn't work because it doesn't agree with what we found experimentally. Dipoles are neutral. So we spent a good deal of time trying to figure out what the structure is, and I'm going to relieve you of the details because I want to discuss other stuff, but I'll just summarize that we came to the conclusion that the structure was something like this, that you have a hydrophilic material sitting next to water, and from that water, these sheets or layers, hexagonal layers, build. And they build one at a time, and here's another one, and so on. And they keep building from the surface. And you can get many layers, as I said, up to millions. And the structure basically looks like this. It's a hexagonal kind of of motif. And if you count the number of hydrogens and oxygens here in one unit cell, it turns out that this is not H2O. This is actually H3O2. You might call it a chemical change because it's not H2O anymore. Now, if this were H4O2, it would be double this one. It would be neutral, and because it's missing one positive charge, it's net negative. And we need net negative to conform to the experimental evidence. And so this is the conclusion that we wound up with. Now, let me switch gears a little bit to talk about the idea of water memory, because water memory is an issue that's been under consideration for many, many years. And most scientists will tell you that this is the biggest bullshit in the world, that water can contain memory. It doesn't make any sense. But other people who come to, especially who come to our annual conference on the physics, chemistry, and biology of water, which has been in Bulgaria, each year is now moving to Germany, present. We usually have 2 or 3 or 4 presentations that demonstrate that water can contain information. And I know that some of you have interest in that. And if you think about memory, of course you think about the thumb drive, and contains information. And what's inside there? Well, it's an array of silicon or silicon dioxide atoms, and they're arrayed not just in this 2-dimensional pattern, but actually in 3 dimensions. So you have a regular arrangement, and each one of these can be addressed independently, and each one has 2 states corresponding to 0 and 1. So essentially, that's what memory is all about. And the hypothesis is that the EZ water has exactly the same capacity. So this is essentially a fixed array, semi-solid liquid crystal array that contains oxygens and hydrogens. And if you could imagine that electromagnetic waves could impact certain of the oxygen atoms, I think this is more likely for various reasons. That, for example, it may impact this one or what have you. If you take into account that these could actually change state, so each oxygen atom has actually 5 different oxidation states, right? So -2, -1, 0, +1, and +2. And so instead of having 2 possible states like a 0 and 1, it's got 5. If you do the arithmetic to figure out the difference in memory capacity of this EZ versus silicon, we did it once. I can't remember. It was something like 10 to the 8th or 10 to the 9th. If I asked Kurt to calculate it, he'd probably do it in a few milliseconds. So potentially, this has huge information storage capacity. And so you can imagine the possibility that this EZ water could be used not only in our body as storage of information, but also eventually as a computer memory. So a question that's related to that is, it's hard to think of liquid crystalline water as inside a computer memory, but imagine if you could solidify it, if you could take this EZ and from it create a solid. And this has been achieved by Vittorio Elia and his group in Naples. And so this is an intermediate result. What he does is he takes a piece of Nafion that's sitting in a petri dish. Well, actually, he doesn't do it. His wife does it. She's really conscientious. They've been married for more than 50 years, and they still hold hands. It's very, very charming. So he has got a piece of Nafion sitting in a petri dish, and she pours some water and lets the EZ build up, and then she takes a roller and squeezes it off, and she does it again, and she does it again and again and again, 10,000 times to get a reasonable volume of this stuff. And then if you take that and put it into a beaker and just let it stand for some time, what you do is you get a clear water, and also you get this cloud-like, which presumably is the solid, the EZs that are coalescing over time, and it looks just like a cloud. And it's no surprise, I think, that you get a cloud-like formation that resembles a cloud, but this is only intermediate. The real result is they take this mixture of easy water and plain old water, and they put it into a lyophilizer, which what it does is it basically pulls off the free water. And this is the result. Here is the flask, and at the bottom of the flask is solid, easy water. And this is confirmed by mass spectrometry to contain almost no contaminants. It's pure oxygen and hydrogen. And this is water. This is some kind of water, easy water, we think, at room temperature. Solid water, imagine, solid water at room temperature. So this is achievable. We had a sample of it, and I feel guilty because Elena spent probably many, many weeks, if not months, to give us this tiny sample. And I sat down with my postdocs and students and, hey, take a little, and see what you can do. And nobody really did anything with it, so I'm feeling guilty. Anyway, this is amazing, and so I think it's amazing. And so a question, will information storage in easy water replace conventional information storage? This is not going to happen in the next month, but I think it's possible that in the future your computer will be filled with easy water. So the answer to question 2 is easy, physically distinct from bulk water. The answer is yes. There are a lot of things that are different, and the best we can understand it, it's a layered honeycomb structure, hexagonal structure, with information storage capability. Now, when you think of information storage in water, who's the first person who comes to mind? Masaru Emoto. Some of you know him? No, I don't see. Okay, yeah, well, okay. So you know that he was a spiritualist who had a big impact, not only in Japan, but all over the world. And his consciousness applied to water. When the water was frozen, it would create different shapes. And this is just one example. So a Mozart symphony played to the water would create a beautiful crystal, and heavy metal, an ugly crystal. And if he said to the water or thought to the water, "I love you," versus "I hate you," the same sort of thing happened. And so he became extremely well known, and even today, even past his death, still various emissaries are looking at these crystals, trying to determine whether, for example, water from a spring or from some company or so produces the good crystals or the bad crystals. So anyway, almost every scientist I know thinks this is nonsense. However, we think there's a possible basis for what they find, because there is the possibility of information storage in easy water. And easy water, we found, is an intermediate between water and ice. You must go through easy water to get to ice. I have no time to talk about that, but it's possible that this underlies what they have found. Okay, now, third question: can crystalline water explain those counterintuitive anomalies? And I'm just going to talk about a couple of them for lack of time. And one was the cloud, and the question was, what's going on here? How come, if the water is evaporating here, just as it evaporates here, is it possible that this water is getting pulled into the cloud or something? Because you'd think that you'd have one long continuous cloud, but sometimes you do, but often you don't. And here I would like to move to a principle that came from a famous physicist I'll mention later. And that is, suppose you take two particles, and suppose they have an EZ around them with negative charge, and this one also has an EZ with negative charge, and also there are positives out here, because if this is an EZ, as the negative charge gets created, you get positives around here. But I ask you a question. If you put two of these into a beaker of water, just take two, and put them near each other so they feel the charge of one another, what happens to the distance between them? Anybody have a guess, or you're too shy to say? They stick together. Okay, everybody agree with that? They both have negative charge, and they come together. You all agree with that or not? Well, you think they go apart. Okay. Everybody agree with him, or do you? Okay. Well, okay. The answer is that they come together. It's sort of counterintuitive because you'd think if they both have negative charge, they must repel. And it turns out there's no violation of standard physics here. What happens comes from the late, great Richard Feynman, Nobel physicist of the last half of the 20th century, who is kind of everybody's hero. And he said, the reason they come together, he said, is because of the principle that he called like likes like. Now, what does that mean? Well, the like charges, the two negative charges, like each other, and so they come together because, you know, when you like someone, you tend to come together. So he said, like likes like, and the reason is because of an intermediate of unlike. That's what he says. So where do the unlikes come from? Well, I think it became clear once we understood about the EZ, because these positive charges are sitting out here, and in the middle between the two you have a contribution from this one and a contribution from this one. So you have them in pretty high concentration here, and because of that, because these positive charges are in between, they attract this negative one and this negative one, and so they come together. And the principle of like likes like because of an intermediate of unlike actually was discovered almost 1,000 years ago, and discovered here in Japan from the Tale of Genji, which was, I'm led to believe, the first novel. And the Tale of Genji talks about different clans that are perpetually at war with one another, and of course they'll never come together unless the appropriate intermediate is placed in between. So like likes like because of an intermediate of unlikes from ancient Japan. So they come together, and the question is, when do they stop? And so it's been shown you get stability when the attractive force, these positives pulling this and pulling this, equal the repulsive force, this repelling this. Then you have stability, and then they're stable. And if you think about a cloud, the cloud consists of these negatively charged droplets, but you'd never think they would get together, but they can get together because of the positive intermediates that sit between them, and they come from the atmosphere, which is full of positive charge. And so you can imagine if you have another one of these negative charged droplets out here, it's going to be attracted by this positive, come together, and the cloud will grow. And that's why you can get isolated clouds that may just keep growing, but nothing around them because of this kind of attraction. So another anomalous phenomenon is this one. This is an old Hungarian coin sitting on water. And I think some of you have had the experience, you put a pin on the surface of the water or a paper clip and it floats despite the high density, but if you put it underneath, it plummets to the bottom. And we wondered whether the usual explanation of the top molecular layer one molecular layer essentially has more hydrogen bonds, and that makes it stiffer, giving you a higher surface tension. We wondered whether that was correct, and we looked at the air-water interface, and we found that this EZ water grows and the top of the water and this interface. So the experiment looks like this. You take two pieces of glass and you seal at the edges here, forming a chamber, and you fill it with water plus microspheres. That's why it's cloudy, because the microspheres scatter light. And so here's the air, and here's the meniscus. And what we found is that within 15 or 20 minutes, a pretty sizable zone without microspheres opens up. In other words, the microspheres get excluded from this top region. And this clear zone looked similar to an EZ because it's microsphere free. And one of the students in the lab stuck a microelectrode near the top and found a big negative electrical potential, just like we find in exclusion zones. And it turns out that the physical character of this clear zone here, it looks like a gel. It's not water. Up here, I'll show you the evidence for that in a moment. It's like a thick rubber band that runs across from one side to the other side. And, and here's the evidence. So you stick a glass probe, touch the surface, and mechanically perturb it and move it back and forth. And what you find is that the thickness of this clear zone hardly changes in a noticeable way. So it looks like this. Oops. Sorry. It's coming down, lifting up, and you see the height of that zone as it moves back and forth doesn't change noticeably. So it sticks together. It's cohesive. And so we learned from this that this top zone is actually like an exclusion zone, has all the characteristics of an EZ. It's not just the top one molecular layer with more hydrogen bonds that is creating this high surface tension. And so we think that the reason that this can float on the top is that there are many structured layers to create the high surface tension. And all of this was an excuse to show this slide, because this slide is a lizard that lives in Central America. It mostly spends its time sitting on the branch, but when it gets inspired, it'll jump off the branch and head for the water and walk on the water. And so because it walks on water, it's known as the Jesus Christ lizard. And the question is, well, does a single molecular layer with a few extra hydrogen bonds enough to explain this, or the thick layer that we see in the laboratory? And I think it's the latter. This EZ layer could grow quite thick. And also, if you have this EZ layer at the top, it's possible to lend support to almost anything that's on the surface, not just that lizard, but perhaps also help to keep boats afloat, over and above Archimedes' principle. Another point is that crystals can be pretty stiff, right? So if you take a crystal like a sugar crystal, a salt crystal, or your diamond in your ring, whatever, they're really stiff and hard. And so if you think about the water bridge, this is so you can compute the stiffness, because the stiffness is a function of the droop, which depends on the distance between here and the diameter of this. And the computed stiffness is so high you can almost walk across it. Water doesn't do this, but EZ water is a crystal, and crystals can be really stiff. So we think that it's possible that the reason for this weird behavior is that it's EZ water, at least in part, that's running from one end to the other, and that's what explains it. So the answer to question 3 is yes. Liquid crystalline water explains many anomalies. Okay, the critical question is what charges the water battery? So every battery needs to get charged, right? And your cell phone runs down, and you got to plug it in, otherwise it doesn't work. And it's the same thing here. And we couldn't figure out what's going on, and we couldn't figure out where this is coming from. You obviously can't plug it into the wall. And it took us, I must admit, in our stupidity, it took us 2 or 3 years to figure out. And we finally got the answer. And I remember I was giving a class once to some undergraduate students, and I asked the same question. We knew the answer already. And I asked, and one student timidly raises his hand, and he said, light? And we already knew the answer, but we quickly grabbed the student to work in the laboratory, because he was obviously came up with the answer that took us a few years to come up with. It's light. And how did we find out? We found out because of a curious undergraduate student who was doing experiments sitting on the lab bench. He's got an exclusion zone next to Nafion, and sitting right next to him was this lamp, a gooseneck lamp. So he took the lamp and he shined it on the chamber. You know, this is a very crude, simple experiment. And this is what he showed me. So this is actual data from a very early experiment, and this is a piece of Nafion. Here is an exclusion zone, and here are the microspheres. And typically the exclusion zone is a long, the edge is one continuous long line, but where he was shining the light, the exclusion zone had grown, as you can see here. And they took away the light, and it basically came back with a time constant of some tens of minutes. So it didn't take a rocket scientist to figure out that, you shine light, it gets bigger, so maybe the energy from the light, the photons, are what is powering the growth of the exclusion zone. Thank you for nodding your head. Okay. So we did a series of experiments to figure out which wavelengths are the most effective. And we went from UV all the way up to mid-infrared. And we found the most effective was actually 3.1, 3.0 micrometers in the infrared. And that could grow the exclusion zone by a factor of 10 easily with a very weak light exposure. So a question, even in this group, who deals with infrared all the time, is, where does infrared come from? So we all know that if we look into the oven or the toaster, the coils are glowing a bright red and it's warm. And we know that the warmth is associated with infrared. So we think of infrared as coming from sources like that. But in fact, infrared is all over. You can't get rid of it. So if we turned off all the lights here, and if I took out not my smartphone camera, which is sensitive to visible light, but an infrared camera, no visible light, I'd get a beautiful image of everything here, no problem, because everything is generating infrared. So this is literally free energy. It's not the chemistry book, free energy, but it's free. It costs not 1 yen or anything like that. So it's always there, which means if you have hydrophilic material next to water, because the energy required to build the EZ is there, you always have some EZ. So EZ is going to be all over the place. Wherever you have the right hydrophilic surface and water, you're going to have EZ. And if you add more EZ, it grows. And if you take it away, it'll come back to the original. So the answer to question number 4 about energy is that EZ buildup is powered by light, and the light creates order. This is ordered water, and also charges the water battery. So what this is suggesting is that this water, which we tend to think is at equilibrium with the environment, is not at equilibrium at all. It's absorbing light from the environment. And if that's the case, then you ought to be able, if this is getting energy from the environment, you ought to be able to capture this energy and do work. So my guess, though, is that nobody in this audience has ever seen a glass of water doing work, right? Or maybe you have. Well, I'm going to give you one example of water actually doing work. And it happened with another undergraduate student who was being disobedient. They like to be disobedient. I'm a very difficult taskmaster. So this student, we just heard that Naphion, I showed you a sheet of Naphion that it came in tubes. So we thought, why don't we start studying these tubes? We wanted to see whether these tubes have exclusion zones. So the student goes into the laboratory, takes the tube, and puts it into a chamber of water. And he confirmed that, you can see EZs building this way and building this way. But he found it was a curious finding. And it was so important to the student that I remember he barged into my office. I had a visitor, and we're sitting and talking. I don't remember if he was an important visitor or someone less important, but the student was excited enough to barge into my office and say, "Oh, excuse me, but I found something pretty interesting." Right? And I'm thinking, is this interesting enough to barge in to my office when I'm having a conversation with an important visitor? He told me, he said, I'm looking with my microscope, sitting down here, looking here, and there are particles in the water so he could see what's going on. He said, I see flow through the tube, and it just doesn't stop. And he said, I thought it might be important enough to tell you. And I'm thinking, this is, if true, and we checked to see that there are no artifacts, if this is really true, this is incredibly important. Why is it important? Because typically if you've got a tube, in order to promote flow, you need to have a pressure gradient, right? So your large arteries, for example, the heart is squeezing, developing pressure, sending flow to those. But here, there's no pressure gradient because this thing is sitting at the bottom of the chamber. The height of this is the same as the height of this, but you see flow. And so I was thinking the only possible explanation for this is what I've been demonstrating to you previously, that the energy from the environment is coming and giving energy to the water, and that energy is somehow getting transduced to produce the flow, which we confirmed. And the experiment looks like this. You just take a tube, fill it with water, make sure there are no air bubbles, and then stick it into a chamber with water and some particles, microspheres. Take it to the microscope and restrict the amount of light by choosing green, for example, which is not so unusual. And this is what you see. So inside the Nafion tube, the flow goes, and we've had it going continuously for as much as a day and a half. And we think we know how to get it extended even longer. So someone said, this must be some special quirk of these Nafion tubes. So we thought, let's try some other tubes, hydrophilic tubes. It's not so easy to find hydrophilic tubes that are small like this. And so we basically created our own tubes. And we did it by making gels. And as the gel was gelling, we had a wire inside. And you pull the wire out, and you're left with a chunk of gel with a tunnel that runs right through it. So you take this chunk with the tunnel, and you stick it in water plus microspheres to see what happens. And so we did this, and so this was a polyacrylic acid gel here and here. Here's the tunnel, and you can see the EZ grows from this edge. It grows from this edge, and it pushes all the microspheres to the center, which is no surprise based on everything I've shown you. And if you look at the video, it flows just the same as it flows in the Nafion tube. We tried a half dozen different gels, and we got essentially the same results. The speed was a little different in each of those gels. So this is a general phenomenon. And then somebody said, we listened to people, someone said, if you think that this is powered by light, why don't you just turn up the illumination and see if it goes faster? Right? Anybody would think of that. So, yeah. And so we did that, and it turned out that we just published 2-3 years ago that light increases the speed by up to 5 times. Just turning up the light, it goes faster. So we wind up with a situation like this, where you have a tube sitting in the water, and you have no pressure gradient there, but yet, since the water is flowing through, work is done, and energy is required if work is done. And so the energy absorption that I've demonstrated to you is a necessary condition, unless some other source of energy could be identified. We haven't found it. So basically, this water is transducing light energy into mechanical energy. Now that seems weird. We never think of water as a transducer of light energy into mechanical energy. But if you think about what goes on in plants, right? So what gives energy to the plant? Well, light gives energy. And remember, the first step in photosynthesis is separating water into negative and positive components, very similar to what we've demonstrated, although what we have is, I think, in more detail than the photosynthesis people think. And what I'm suggesting to you is that the same thing happens in water. And in the plant, gets its energy and it powers growth, metabolism, bending, what have you. And I'm suggesting the same thing occurs in water, that the water is a transducer, which transduces light energy into, in this case, mechanical energy, but we found other kinds of transduction. So you wind up with something like this, which physicists will recognize as looks similar to an iconic equation that they know about, E equals H2O. And of course, the units don't match, and for that, I apologize. But I think you get the idea that this water is actually containing energy, contains potential energy to drive work. So, okay, so why is all of this important? I think that it's foundational for any or all science involving water, molecules, and light. And what I want to focus on is for your health, your health, my health too. And so the question is, does human biology use radiant energy? Right? And so here we are, receiving a light energy all the time, and some of it penetrates some depth. And the question is, do we use it like plants, or do we not use it? Now, if you were Mother Nature and you were pretty successful with green plants, you have an energy transduction mechanism that works pretty well, and you decided, hey, it's time to invent animals. And animals can kind of move around and get food and get their energy from food. Would you throw out a process that has been so effective for green plants and some bacteria, or would you sort of keep it in reserve? Bernard says he'll keep it in reserve. So if he says so, it must be correct. So the first idea is maybe the cardiovascular system, because you got capillaries that are pretty near the surface, and they certainly receive energy from light energy from outside. You can even, when you're dark adapted, you can put a flashlight here and see it coming through your hand. So some wavelengths penetrate pretty deeply. And I first got this idea from a Russian. I went to visit my friend Vladimir Vyakov in Moscow University, and the first thing he does is, you got to meet my next door neighbor in the laboratory next door. So the guy comes marching in, and his English is not very good, and my Russian is zero, so he somehow translated. And the guy said, there's a big problem in the cardiovascular system. And I'm feeling pretty arrogant, because my PhD was done on simulating the pressure and flow in the cardiovascular system. And I thought for sure we had all the answers. So what's this guy going to tell me about a big problem in the cardiovascular system? So he said there's a big problem because, the red blood cells are 6 or 7 micrometers, but the capillaries through which they flow, some of them in healthy young adults, unlike me, are not 6 or 7 micrometers, but 3 or 4 micrometers. So, at first glance, it looks like nature made a big mistake, because there's a big plumbing problem, to push these red blood cells through these narrow channels. Did Mother Nature screw up in some way? So he said, this is a big problem, he said, because you got to squeeze these red cells through, and to do that they have to kind of bend like this and push them through. And he calculated the resistance of this, and it turned out that his calculations showed that if the heart itself, the pressure of the heart, were responsible for driving the blood through these capillaries, that the heart would need to develop something like 1 million times the pressure that it actually develops. Impossible. Even if he's off by a couple of orders of magnitude, it just can't be. So so he came up with a few theories, and I'm thinking, we get this spontaneous or self-driven flow through these tubes. Maybe the same principle could apply inside your capillaries and my capillaries. And so this one illustrates the problem. This is a piece of muscle tissue, and here are some capillaries running through, and you can see the red blood cells. They're supposed to look like this, but you can see they have to get squeezed in order to go through. So some of them struggle mightily to work their way through. But anyway, this is a kind of unrecognized problem in the cardiovascular system. And I'm thinking, we see this flow here, and it's driven by radiant energy. And, it's kind of self-evident to raise the question, might radiant energy help drive blood flow through your capillaries and my capillaries, maybe assisting your heart do it? And we had some interesting background material to suggest that maybe there is something like that. We had found out about an Israeli group, and they were studying blood flow in mice. So they put the mice out, they open the chest, they do their experiment, and after the experiment is over, they clamp the aorta, and the mouse complies by perishing afterward. But they noticed they continue to measure the capillary blood flow using optical coherence tomography. It doesn't really matter, but this is an effective method. And they found that the flow didn't stop. You expect when you die, or when the mouse dies, the flow stops, but it didn't stop. It actually it was a lower velocity, substantially lower, but it kept flowing. And so they're looking at their watch, 5 minutes, 10 minutes, 30 minutes, 1 hour, just kept going. And they tried this in 10 different mice, and they found the same result. And they started scratching their heads. They couldn't figure out what's going on. It turns out that there were 3 or 4 other groups over many years who have found the same phenomenon. So something interesting is going on because there's something else besides the heart that's driving this flow. We never think about it. So a student in my lab, Li Zheng, has been studying this phenomenon, and he studied it in the chick embryo at 3 days, where the neural system and the feedback systems and such are not yet well developed. So here's the heart and some vessels, and he's measuring here or here. So here's the eggshell, and you basically just cut off the top of the eggshell, which reveals the circulation. And so the first thing he did was to put KCl in the heart, and that stops the heart right away. So the question is, does the flow continue or doesn't continue? And he found, this is flow versus time. He found that flow was reduced greatly at the time the heart stopped, but it continued to flow. And then the critical test to see if our mechanism applied the signature characteristic is you apply infrared and it gets faster, and you can see here that when the infrared is applied, the velocity increases by about 3 times, and it's reversible. So we think it's possible that in all of our cells or all of our capillaries, that this phenomenon may be applying, that the infrared energy is actually propelling flow in your capillaries. So where does the infrared energy come from? It could certainly come from externally, but your metabolism is generating heat. The heat is essentially infrared energy. So it's possible that your metabolism itself producing heat, that heat is not just radiated out, but some of it is used. It's used to propel flow in your capillaries. So now, the question is, if this works in capillaries, what about the rest of your body? Is it just the cardiovascular system, or is it possible that we use light in other ways? And this is something we don't really think about, and I think the answer is yes, that we do. So think about any cell. Okay, so you got a cell, and it has a pretty yellow membrane in it. And inside the cell, you've got water and you've got macromolecules, most of which are proteins, and proteins have charged surfaces. So you've got water next to charged surfaces like this, you'd expect to have exclusion zones with a negative charge, which you can see here. And the real cell, of course, is so crowded that essentially all the water inside the cell, or almost all the water, is going to be EZ water. And remember, just as a side issue, but EZ water is negatively charged. And if you stick an electrode in the cell, you measure negative charge. All cells are negatively charged. And of course, we read in the textbook that this is caused by some membrane components, pumps and channels and such. And when I was a student, I learned that, and I could never understand it. I just thought I was too stupid. But I think a simpler explanation, and it's a side issue, so I won't go into the evidence for it, but a simple explanation is the cell is negatively charged because it's filled with interfacial or EZ water, which is negatively charged. That's simple. So basically, you've got a lot of negative charge that sits in the cell. Negative charges repel one another. They want to get away, far away from each other as possible, and this is essentially potential energy. And this potential energy could be, and I think is, used to help the process of protein folding. And protein folding, as you know, is what does the work of the cell. So in a muscle cell, the muscles contract because the proteins, the myosin, actin, fold in a certain way, and then they unfold. And so I'm suggesting that the energy for that could easily come from this high concentration of negative charge. So it looks something like this. It looks like you got a protein, and the protein is surrounded by EZ water. And what happens is the EZ water melts, and the protein then folds, and then it unfolds. And as it unfolds, the EZ water builds back up again, and your at the initial condition. Now, if you have a problem where, on the right side where you don't have easy water, or you don't have enough easy water, basically the cell is dehydrated, then the proteins can't function in the same way because they're out of their usual environment. They misfold or don't fold or whatever, and you've got a pathology or a non-functional cell. So now if you think of, if you connect the dots about the energy and where the energy comes from, remember, light builds the EZ, which builds this negative charge, which contains potential energy, and this potential energy does the work of folding of proteins, which is essentially the work of the cell. Now, so essentially, if we go follow the arrows from here to here, light is actually doing some of the work of the cell in our cells. So when you ask, where do we get our energy? The first one, the obvious one, is food, because it's clear that we get energy from food. But not recognized is that we also get energy from light. The light is absorbed by the water, which builds EZ, which delivers energy to our cells. So we get some energy from light, just the same way as plants get essentially all of their energy from light. Now, should this matter to you? Well, water matters and light matters. And so the question you might be asking is, for your health, you need to make sure you're hydrated. You need to make sure you have enough easy water inside your cells. So what can you do? What simple things can you do to maintain your health? And I'm going to list 6 of them, 6 that have been known pretty much throughout the ages, but we haven't really paid much attention to them. And I think it's possible that all of these 6 simple things you can do all are centered around buildup of easy water, because you must have easy water in your cells to function properly. So the first one is drink a lot of water, or enough water. It's the raw material for building easy water. So you need to have enough of it, especially when you're sick. And the second one is green juicing. So what is green juicing? You take a freshly grown plant and you squeeze the hell out of it, you squeeze the juices. And what are the juices? The juice is the inside the cell, the water from inside the cells of the plants. And they contain lots of easy water. So basically, you're drinking what contains, and we've confirmed it contains lots of easy water. And you drink it, and basically it replaces the easy water that may be missing from some of your cells. So it promotes health. And in consulting with various alternative, so to speak, practitioners, they tell me that the first thing they do when a patient comes, it doesn't matter what the problem is, they tell them to do this green juicing every day. And the report is that no matter what their problem is, they come back in a few months, it's better. And I think this is possibly the reason. Another one is various substances that have been known throughout the ages to be good for health, back from Ayurvedic times and ancient Chinese times. And some of these are known to be generally good for your general health. So what's going on there? Are there multiple receptors for these things, or is there possibly one central mechanism? And we're thinking that some of these, a few of them are listed here, maybe what they tend to do is build easy water. And we did the experiments, we just published, and so turmeric, for example, has curcumin, known for 5,000 years or more. We found in concentrations that would be common inside your body, they expand the EZ. We found the same for coconut water, coconut oil, ghee. We tried basil and several others, even salicylic acid, aspirin. They all expand the EZ at the concentrations that are relevant. And now another one is sunshine. In Seattle, there's not much of it in the winter. When the sun comes out, we look up and we see it and we feel good. And of course, possibility is much of this is psychological, but when the sun comes, it contains some infrared. Infrared builds easy water. It might be that you feel good because the sun is building easy water. And the extreme of that is sauna. It's hot. The heat is associated with infrared. We absorb the infrared. And the infrared builds easy. And if you start with sore muscles and a headache, you come out 20 or 30 minutes later feeling much better. Possibly, this is simply because it builds easy water in your body. And finally, earthing or grounding. When you connect yourself to the earth, what you're doing is connecting yourself to a source of negative charge. I didn't know this until 10 years ago, but a lot of you know it, that the earth is not neutral. It's negatively charged. So if you take off your shoes, and step on the earth or on the grass or on the sand, you're actually electrically connecting yourself to a practically infinite source of negative charge. We found in the laboratory, if we add negative charge, EZs build. It makes sense because EZs are negatively charged. So by connecting yourself to the negative earth, you're absorbing electrons, and these electrons are building EZ water. So these are six different expedients that are simple, that you can do, that are known for many years to improve health. So I'm not suggesting that we photosynthesize, but I am suggesting that we use light in ways that I've explained. And I'm going to end, and I see I've got 4.5 minutes left, with practical applications. And one of them that Kurt is working on is getting energy from sunlight and water, right? We have this charge separation here. Should be able to light a light bulb if you put 2 electrodes in here, and Kurt has demonstrated this very nicely. These are some chambers containing naphion and electrode pairs. This is a switch, and underneath this magnifier is an LED, and Kurt's hand is there, and you can see that it works. And so the idea is now to build this up in such a way that it becomes practical. And this is just using water and light. It's entirely renewable, getting electrical energy. And another one is getting drinking water from contaminated water. And we have a company called Fourth Phase that is actively working on this development to make it practical. And it's simply based on the principle as shown here. This is a naphion tube, and flow comes in, and the flow comes in with not only water, but it contains all the junk that you can imagine, from pharmaceuticals to bacteria, to whatever. And I've shown you that there's an exclusion zone that develops here, and it pushes all the junk into the center. And for typical junk, representative junk, we use microspheres. So we developed the so-called differential extractor, which is a fancy name for 2 tubes, 2 concentric tubes. So the second one, the center one, collects the microspheres and dumps it, and the outer ones here, or outer one, collects EZ water, which comes out here. And we've been able to get a separation in a single pass with this prototype model of 200 to 1 separation. And the question that we're raising now is whether it's possible that salt is also separated. It might be. We're not sure. We've been pursuing this. And if it really is true that we can do that, then what we're doing is, without any physical filter, we're just using the energy from light to create this kind of separation. It's possible that we can put ocean water in here and get drinking water out of there, and the drinking water will be EZ water, which should be healthful. So conclusions. Water, we think of water as having 3 phases: ice, water, and vapor. And I've demonstrated to the evidence for a 4th phase, this EZ water, or interfacial, whatever you want to call it. And it has a structure that's very similar to ice, but not the same as ice. And we found experimentally, no time to show, if you want to freeze water, you must obligatory pass through this phase in order to get to ice. And if you melt the ice, you must pass through this phase in order to get to water. And this has broad implications. The first, I guess the most central one, is this is absorbing radiant energy all the time. And I've given you an example of a biological application in blood vessels. In chemistry, the explanations for reactions that take place in aqueous solution, you've seen it, you've read it, sometimes they're complicated. None of these take into account any of the features that I've demonstrated here. If these features are correct, maybe they're not, if they're correct, then many of the interpretations of basic chemistry need to be revised. Weather, we never know whether to take an umbrella to work tomorrow or not. And the reason is the forecasters don't really know how to predict the weather. They do it with some reasonable accuracy, but they talk only about temperature and pressure. I've never heard the word charge used in any weather forecast, never, not once. And it's obvious that clouds contain charge. Sometimes you see obvious manifestations of it. And I think that with an understanding of the role of charge in these clouds, a lot more understanding of weather will come. For health, I've given you a few expedients that I think are simple and important, involving easy water. If you want to either freeze food or dehydrate food, it's nice to know something about the water. I've shown you practical applications for filtration, possibly desalination, and getting electricity. And before my very last slide, I'm going to mention this Institute for Venture Science. This is not related to what we're doing in water. It's an institute that we created to ultimately fund promising ideas that challenge conventional thinking, that ideas that may have the capacity to bring scientific revolutions, of which there have been very few fundamental revolutions lately. And we got more than 200 pre-proposals. We evaluated them. We picked 5 exciting projects, and we're now looking for donors to help fund these projects. So we're excited about this, and if you know anybody who has done very well and would like to give something back to society, please have them contact me. And finally, what I've shown you here is only a small fraction of what appears in this book. And so any of you who are interested, please have a look. The Japanese translation is not quite ready, but is coming pretty soon. It just needs some editing. So I stop here, and it looks like I have 1 minute and 17, or am I interpreting wrong? No, opposite. Anyway, and so if there are, I don't know, 1 or 2 questions, but I think everybody's hungry, so I probably will quit here. Anyway, thank you.
Well, thank you very much, Professor Pollack.
Sure.
As always, very inspiring.
Thank you.
So we do have, I think, questions. So Professor Palushev, please.
Mr. Pollack, I am a representative of a small group of scientists which try to explain all these phenomena and much more phenomena in other systems on the basis of new theory called torsion field theory. This is fifth fundamental interaction, which give better explanation of many of these phenomena which you mentioned in your report. For instance, easy water, disordination, but you mentioned the notion of information. Impossible to use such a notion when you want to explain, well, the easy water or other phenomena on the basis of electromagnetism.
On the basis of?
Electromagnetism.
Oh, okay.
All explanation here in this conference, I saw, are based on electromagnetism, one of these 4 fundamental interactions. We introduce something else, another interaction, fundamental interaction, which raise our possibilities, our potential to explain many things. Tomorrow I have
Okay, okay. Yeah,
report, and
yeah.
I shall mention about other applications, but especially in water, I can mention that many anomalies, the capillary phenomena, which the capillary phenomena are the base of the plant life. Without this phenomena, this movement of the water, anti-gravity movement against the gravity, it is because of some additional energies which electromagnetism cannot expose.
Yeah,
And many others. For instance, this strange behavior of the water between, in the temperature interval between 0 and 4, and many others.
Thank you. I use the term electromagnetic energy loosely. Nobody really understands, and it may be some kind of energy well beyond electromagnetic energy that we don't understand at all. So, for example, I just give one example of Serge Kernbach, a German engineer scientist who uses impedance spectroscopy to measure in water. And he sticks his electrodes in and measures the impedance spectrograph, and it's pretty consistent. And then he asks somebody from 10,000 kilometers away to focus their attention on the water in his laboratory. And after 30 minutes, he makes another measurement, and the result is completely different from what he had observed before. So there's some kind of energy that nobody understands. Maybe it's not electromagnetic energy, but nobody understands that's creating this. But he's not alone. There are many people who demonstrate. For example, Luc Montagnier won a Nobel Prize. He can demonstrate you take energy from DNA, or better, from the water that surrounds the DNA, and that information is enough to create new DNA that has the same sequence as the original DNA. So nobody understands what's going on here. It may be electromagnetic energy, but it may equally be some kind of energy about which we're completely unfamiliar. So I'm with you on that.
Okay, well, I believe we ran out of time. So if you have more questions, there will be banquet, and you can ask Professor Pollack. Thank you very much for this very nice.
Thank you.
I'm sorry, it's Professor Julian.